Mosquitoes function as highly specialized biological machines, perfectly evolved to deliver specific pathogens directly into the human bloodstream. Within the foundational anatomy of the class Insecta, the most conspicuous feature of this machine is the forward-projecting proboscis. A distinct fringe of scales is located along the posterior margin of the wings, serving as a key diagnostic feature for the family. The biological imperative is strictly divided by sex. Only females take a blood meal to support egg development, while males survive strictly on plant sugars. These specific feeding behaviors and morphological traits form the absolute foundation of epidemiological disease transmission. Public health classification begins at the phylum Arthropoda to track these vectors accurately. This taxonomy branches medically important species into two primary, distinct genera: Anopheles and Culecinines. Within the Culecine branch, subgenera Culex and Aedes are the species of primary medical concern. Each genus serves as a biological vector for different pathogens, such as malaria in Anopheles versus Bancroftian filariasis in Culecines. Rapid visual diagnostic differentiation between these genera is the crucial first step for health professionals responding to a localized outbreak. We can establish a comparative diagnostic framework to evaluate Anopheles against Culecines side by side. The fastest macroscopic diagnostic tool in the field is observing the adult resting posture against a flat surface. The body of the Anopheles points at a sharp angle away from the wall. The Culecine rests nearly parallel to its resting surface. Microscopic diagnostics focus on female head anatomy, specifically directing attention to the maxillary pulps. In the female Anopheles, the pulps are exactly as long as the proboscis. Female Culecines have drastically shorter pulps. This palp-to-proboscis ratio is the definitive identifier for dangerous blood-feeding females. Male mosquitoes require different identifiers since they do not possess blood-feeding mouthparts. Diagnostic attention shifts to the male antennae and the shape of the extreme tips of the palps. The male Anopheles features distinct club-shaped tips on the palps. The male Culecine has bushy antennae, but the palp tips are not club-shaped. These differences begin at the start of the life cycle with oviposition, or the pattern of eggs laid on the water surface. Anopheles lay single eggs equipped with specialized lateral floats. Conversely, Culex species bind their eggs together in a floating raft, while Aedes lay eggs singly on moist substrates. These specific egg morphologies dictate which water bodies health workers must target during early surveillance. We can now apply this diagnostic matrix to specific real-world threats, starting with the major malaria vector in Sri Lanka. Anopheles culiciphates is a small, brown mosquito characterized by banded wings and banded pulps. The primary vector of Bancroftian filariasis is Culex quinquefasciatus. CX quinquefasciatus is also small and brown, but it lacks any distinct body markings on the thorax, wings, or legs. Aedes species constitute the final medically important profile. These species feature highly distinct black and white stripe markings on the thorax and legs. Physical profiles correlate with unique feeding schedules. Aedes, for example, typically bite during the day. Daytime feeding contrasts with Anopheles and Culex, which are strictly nocturnal hunters. Identifying the vector's physical profile immediately establishes its specific behavioral timetable for public health workers. Analyzing the adult mosquito within its complete holometabolous life cycle reveals clear points of environmental vulnerability. Larval habitat preferences vary by species, which dictates the environmental control strategy. Anopheles require shallow, sunlit, and clear, slow-moving water, often found in gem pits or cattle hoof marks. Culex breed profusely in dirty, organically polluted water collections, such as stagnant city drains and leaky septic tanks. Aedes exploit clean standing water in artificial containers, like vases or tires, both indoors and outdoors. Source reduction must be precisely tailored to these specific ecological niches to be effective. Eliminating the correct breeding ground stops the disease chain before the vector matures into an adult threat. Adult behavioral traits distinguish between endophagic, or indoor, and exophagic, or outdoor, feeding and resting. Both Anopheles caliciphaces and Culex quinquefascites are endophagic, resting on indoor walls after taking a blood meal. This resting behavior makes them vulnerable to indoor residual spraying, or IRS. Because these two vectors are nocturnal, physical barriers like bed netting are highly effective defenses. Daytime exophagic biters like Aedes bypass these indoor defenses, requiring personal repellents and protective clothing. Effective disease control is a battle of specific intelligence rather than a one-size-fits-all approach. By combining precise morphological identification with behavioral and ecological data, public health professionals can design targeted interventions to break the chain of vector-borne diseases.